inflammation. So it's gram-positive cocci accumulate as pus. So you can see dead and dying neutrophils there and organism also there. So there are virulence factors. So as I said earlier, bacteria should adhere, go for the colonization. Now body also act against those pathogens. But at the same time, if bacteria having multiple disease causing capacity, armory, so bacteria will cause the disease. They win the battle initially. So they produce enterotoxins. There are cytolytic toxins, enzymes, and protein A. So enterotoxins, we can classify as heat stable and heat labile. These are endotoxins. So part of the bacterium cell wall. So protein produced by bacteria released into the environment. They are heat stable. So they lead to food poisoning. That's another way. And toxic shock syndrome. That's another one. And pseudomembranous colitis. So they cause toxic shock syndrome. That is superantigen mediated. So toxins are there. So even in the absence of bacteria, when the produced toxins are there, these toxins lead to toxemia. They reach into our body circulation and cause inflammatory reaction and cause for sepsis. There are other toxins like exfoliating or epidermolytic toxin. So they lyse our epidermis, and this is one toxic shock syndrome, toxin 1. Then cytolytic toxins. They affect red blood cells, so they lyse the cells. Cytolytic. So they mainly lyse erythrocytes and leukocytes. There are hemolytic toxins, what I said earlier, alpha, beta, gamma, and delta. Then another one is pentan valentine leukocyte. Oh, it is lethal to white blood cells. This is mainly produced by community-acquired methicillin-resistant Staphylococcus aureus, but it is there in the Staphylococcus epidermidis. So PVL, we say, it's another entity. Then enzymes. Hyaluronidase, they act on hyaluronic acid, they are part of extracellular glycosamine glycan and matrix, they lyse. Staphylokinase, so it acts on fibrinolysis, which allows spread of infection. They cause lysis of the fibrinolysis. Fibrous is the clot, fibrin clot. Coagulase, it's a virulent marker as well as it's helped to differentiate acid Staph aureus or coagulase negative. Lipases there, this acting on lipids present on surface of the skin. At the same time, there are some enzymes, these penicillinases. They destroy penicillins. Then DNases, they degrade DNase. So then this is an enzyme which produce penicillin resistance. Penicillin is the chemical molecule is beta lactam. With the presence of beta lactamase, they destroy this. So protein found in cell wall, they bind to Fc part of the Fc portion of the IgG antibodies and blocks phagocytosis. It's another protein which prevents phagocytosis. Phagocytosis is with the presence of bacteria or foreign agents, phagocytes. So macrophage will come and act on these, and it's a kind of absorbing them, swallowing like, so it is prevented. So clinically, I said it's a skin and wound infection. Clinical infection, skin and wound, impetigo, they cause furuncle, boils, they act on our hair follicles, usually in areas of highly sweating areas. So if they act as carbuncles, it's a cluster of boils. So single one is furuncle or boil wheel. It's infected one hair follicle. If it is multiple and they coalesce together, it is termed as carbuncles. And also it is called surgical wound infection as well. So scalded skin syndrome, another entity. It's common in children. So extensive exfoliative dermatitis, young children and newborns. It's termed as Ritter's disease. And toxic shock syndrome, multi-system disease caused by toxic syndrome toxins, affect mainly women, children, also men. Food poisoning, I said, there are enterotoxins, enterotoxin A, which cause mainly. The effect include meat, dairy products, so they are there. So source is infected with the food handling. Other varieties, coagulase negative Staphylococcus, found as indigenous. Said earlier, it's a normal flora. So usually, if we found in the samples, we think it is contamination. But in immunocompromised or patients with long-term indwelling devices, risk of infection is high. So it habitat in skin and mucous membranes and common human isolates, I said, epidermidis, Staphylococcus saprophyticus, and hemolyticus. So it's a predominant hospital-acquired infection. So skin flora get introduced by catheters, heart valves, CSF shunt, produce the swim layer and helps adherence to prosthesis and avoidance of phagocytosis. So they produce biofilms. So UTI, so common entity, or at the same time infective endocarditis is there. So prosthesis device like prosthetic cardiac valves, they're more prone to infect. At the same time, prosthetic joints, knee hip replacement, they produce late infections leading to osteomyelitis, septic arthritis. So other what I said is young sexually active women, saprophyticus is a common entity. So we have to do urine culture once patient comes with dysuria, fever, maybe not. Urine frequency, blood in urea, that is hematuria. So we do a urine full report. At the same time, we do a urine culture. And depending on the clinical scenario, if it's severe, we start antibiotics. Otherwise, we can wait. And if we isolate these organisms, we have to treat. Usually, we treat with the oral antibiotics. If it is severe case, we go for intravenous. Hemolyticus, it's another organism, coagulase negative one, inhabited in skin and mucous membranes. It's rarely cause infection, but associated with wound infection, bacteremia, and endocarditis as well. So we'll discuss a bit about laboratory diagnosis. So laboratory diagnosis. So when the patient develops symptoms, we always go for the laboratory diagnosis. So there are investigations. We'll do full blood count, C-reactive protein level, assess the inflammatory marker. It's an inflammatory marker. Then, if the patient having a focus of infection, we'll say skin. We try to take a sample from there as well. At the same time, if patient is having fever, we take a blood sample for culture. Usually, cellulitis, erysipelas, even necrotizing fasciitis, blood culture positivity is very less, less than 10%. Even the skin, we can take a tissue culture. Swab is not advised. If vesicles are there, we can rupture and get the fluid. Otherwise, always advice is when the patient is at the operation theater doing a wound debridement or wound toilet, we can take deep tissue samples and go for the culture. So depending on the clinical scenario, if patient is having pneumonia, you have to take a sample as from the disputer, nasopharyngeal aspirate. If patient is having urine tract infection, suspecting, we take midstream clean catch urine. Likewise, if it is we suspecting endocarditis, we have to take a blood sample. If patient is suspecting gastroenteritis, we have to take a vomitus is there, we can take a vomitus. At the same time, diarrheal sample. Likewise, depending on the focus of infection, we have to take the sample. And if the patient is having fever, always blood culture is advisable. So collection, we have to take a sterile containers always, because if it is other organisms there, it may contaminate, and we are nowhere there. We don't know is it real pathogen or not. So sterile means no organisms, no spores, nothing there. So that's how we have to take that's the container. Then we have to properly label. We have to wear the gloves under always without any contamination. We have to take the sample. Once labeling is there, we have to label. That means with the bed ticket number or clinic number. Other characteristic include the container is it should leak apart from sterile, leak proof one. Otherwise, if it is leaking is there by direction or one way, it can contaminate the surface and surrounding and all the area. Other way, it can sample it can contaminate from foreign or outside pathogens or microorganism. Again, nowhere there. So then once the labeling is done, we have to keep them in, we have to transport them in appropriate condition. So usually we have to transport in to the laboratory. Otherwise, we can anticipate the delay. We can keep in the ward setup in the ward and or theater and send there. So once about the laboratory diagnosis, we can do direct smear examination. So once swab is there or pus sample is there from the skin and soft tissue or vesicular fluid, what I said, or maybe tissue sample. So we can do gram stain and see if organisms are there. Here you can see gram-positive cocaine clusters. We can think possibility of infection. So neutrophil also can be seen here, polymorphonuclear neutrophils. Usually in the pathogen, there are neutrophils also there. So without neutrophils, only microorganisms there, we can say probable contamination. So it provides the early clue with the direct smear. At the same time, we go for the culture. So we do culture in blood agar, Mekun agar, and we put them blood following inoculation, we put them in the incubator. So we want, our ultimate aim is provide the good support, good media, good nutrition requirement for them to grow them. See, mimicking in the tissue, like in the body. Body, there are a lot of antibodies. So maybe earlier taken antibiotics, but here those are free. So it's a favorable condition to these microbes to grow. Our aim is we want to identify the pathogen. So we have to provide